So this is a cool article. The scientist is just kind of this random science, kind of somewhere between everyday kind of person science and real science, like hard science and stuff. But goes in the genome, and there's all kinds of new stuff coming out about epigenetic changes and how your life experiences are recorded in your epigenetics that then are passed on to future generations, and that your experiences could be influencing as far down as even your grandkids or even your great-grandkids and stuff. So yeah, think twice about that funnel. No, I'm just kidding. I don't know. They haven't really established the hard connections between in humans yet, but yeah, there's it's pretty cool. It's I always knew there must have been. Back in the day, say 20 years ago, I said, I want to study how the environment is recorded in your genetics. And I was told I was crazy. So, apparently not. So, all right, couple of announcements. Here we are. Last time I'm actually just talking to you. So, if DOS, I forget what it stands for. Dean of Students. Dean of Students office. So you got a bunch of exams and you're rescheduling the final. You have to go through them. I have absolutely no control over whether or not you can or cannot. I had to skip, when I was an undergrad, I had to skip a family trip to Ireland because I had one final. My mother-in-law paid for tickets, paid for cottages on Inch Beach for 2 weeks, and I had one final that they would not allow me to reschedule, and I had to miss that 2-week trip to Ireland because of it. And so I have no control over it. But if you do have a bunch of finals, you can get the Dean of Students to approve you to move your final. If you do that, you need to do that like soon, like today and tomorrow, so that you can work out when you're actually going to take the final. Same thing if you have CST accommodations. Dr. Malone, that's the other section. So don't contact Dr. Malone. He won't know who the heck you are. Okay? Class help hours are today. This is the last help hours for the semester. So you will have a chance to talk to your TAs, and I'll talk about that in a second after this time. But today, 4:30 to 6:30, the TAs will be available to answer any questions you have about any material, whether exam 3 or final, whatever, or free game. You can, whatever you got trouble with, bring it there, and they can help you with it. And so we do have the exam on Wednesday on these chapters. That's mutation, molecular, genomics, cancer, quantitative, and evolution. Then the final is us, 9 through 12, so next Monday at 10:30 here. So the exam is Wednesday. We'll be grading the exam on Wednesday, and we will get it back to you for you to look over in section on Thursday, Friday, whenever you have section. After the exam, I will post on Husky CT exam 1, 2, and 3 from another semester with keys, so that you're not just left out in the cold. You have something to work with, something to practice with, the types of questions that I tend to ask. And then in your section, you'll take a quiz, your last quiz on quantitative genetics and evolutionary genetics, on the stuff you covered last week. You'll go over your exam 3, and then any remaining time, the TAs, that will be your kind of last chance to really kind of hit the TAs up for some extra help on any questions or clarifications that you might have. So if you're still, after, say, Wednesday's exam, you're still having trouble working something out, you will have another opportunity during your discussion section this week to ask your TAs general questions about the material. Don't forget, student evaluations. You'll have an evaluation for me. An evaluation should have one for your TAs, though that has not worked for the last 2 semesters for some reason. But I encourage you to fill them out. I do read them. I do try to improve the class each semester. I would like to say I'm sorry about the honors component. I wasn't able to do all that much interesting stuff for the honors component this semester because right 3 days before the semester started, they yanked my TA funding for the honors component, and so I ended up shorthanded for TAs, and so I wasn't able to do more interesting things for the honors component. So aside from that, I know that needs improvement, and it will get improved in future semesters. I didn't have time this semester. And then also don't forget to thank your TAs. They put in a lot of work. They are overworked, underpaid grad students who have their own research that they need to accomplish. They have their own classes that they're taking with their own exams that they're studying for, and they take all the time to do all of your stuff and grades and all of that kind of stuff. So they put in a lot of work, and so it's worth at least acknowledging that they are, that they do put in a lot of work and are a key component to the course. So, any general questions before I start? What I'm going to do is kind of go back to the beginning, but I'm going to try to tie it forward to the stuff we're talking about for exam 3. Kind of give you a little bit of review for the final, but at the same time kind of show how it connects to exam 3 and how everything's all on the same page. But any general questions about anything? No? So, central dogma. We talked a lot about central dogma. So there's a slide for, I think, slide for every lecture for the molecular genetics section. So one thing I just to kind of orientate ourselves at where we've been and what we're doing. So remember what the central dogma is just essentially saying is that the genome is where we store our information. That's where the information is stored for all living organisms. And so ultimately, what makes one living organism different from any other is how that information is converted into meaningful functional information within the actual organism. And so that is what we mean by gene expression. And so that's that process of transcribing RNAs. And so we talked to death about the molecular aspect of transcribing RNAs, but ultimately what that means though is the transcription of an RNA means a gene is on, okay? And so often what we're going to be doing in section 3 anyways is applied. If we're talking about RNA transcription, the reason we're interested in RNA transcription is because there's a whole lot to both a healthy state and a disease state that is in the RNA information. The genome is important, and so we talked about the genome and the importance of the genome. We talked about mutation, like how we looked at how a mutation in cystic fibrosis gene can cause cystic fibrosis, how you can get that expanding trinucleotide repeat in Huntington's disease, and that's the Huntington gene that gets, and so that one gene can give you Huntington's disease. But beyond those basic gene to phenotype, gene to disease state, a lot of what is really the nuts and bolts of what makes an organism is this step here of taking that information and putting it into an RNA. And so often what we're looking at is RNA levels, because that's going to tell us what genes are on. We have 24,000 genes. We don't always have those genes on in every cell type. Okay, so different genes are going to be on in different cell types at different stages of development in response to different environmental stimuli. And so understanding what genes are on when is an important thing to know. And so what we're able to do with this is to identify, using that information, we're able to identify the disease-causing genes, okay? So such as looking at, say, healthy lung tissue versus a lung cancer tissue. When you look at those two tissues side by side, the reason you look at them side by side is by looking at the healthy lung tissue. This is telling you what a normal lung tissue needs to operate normally in its daily function, okay? And so that gives you the baseline. That's what normal lung tissue looks like. And then you look at cancer tissue and look at the expression values of cancer tissue and what genes are on, what genes are off, okay? And that's going to tell you what genes are potentially contributing to what made that healthy lung tissue into cancer tissue. Okay, because cancer tissue is derived from whatever tissue it came from, whatever organ it's in, that's what tissue it originated from. And so something changed in the original tissue that made it stop being lung and became cancerous lung tissue. And so this understanding of what's at the RNA level, this is what we're looking at when we're talking northern blots, microarrays, RNA sequencing, all of that stuff is looking at that expression level and that differential expression level between different states, okay? Ultimately, we talked about how this is oversimplification, that there's a whole lot going on at the RNA level, because I mean ribosomal RNAs, tRNAs, siRNAs, microRNAs, and a whole bunch of other types of RNAs we haven't even talked about that never get to the protein level, but obviously the protein level is important, and you need to know how we translate from RNAs into protein. We need to know about, we talked about how we process our RNAs and how we process them before we export them out to the cytoplasm. And so we talked all about how we can regulate this whole process from going from housing the information to actually manifesting that information, and how different, and if you interrupt that process, that's how you can sometimes result in things like a disease state. So going way back to the beginning of the semester. So this figure, I like this figure because it really illustrates a lot of different points. So we started the semester out way down here, looking at this macro scale, looking at chromosomes, looking at the way chromosomes divide in cell division. And we looked at how, at the macro scale, how you could get chromosomal rearrangements, these large deletions, inversions, insertions, translocations, all of these things at the macro scale, of large scale chromosomal segments being rearranged or lost, okay? And then we looked at how that can result in disease and different types of, outcomes of that kind of stuff. But one thing to always keep in mind when you're thinking about, when you're looking at a chromosome, is that chromosome is one long linear piece of DNA, really long linear piece of DNA that's made up of individual nucleotides. And so we spent a lot of time looking at this and the way that chromosomes act in a cell during cell division and meiosis and mitosis and all of that stuff. But then we spent a whole lot of time up here looking at the structure of DNA and the basic structure of DNA and how we're using and I'll talk more about structure in just a second, how we use the DNA as a template to either make new DNA or to make RNA. Well, all of those general concepts of DNA, of the molecular genetics is the principles that we found all of that applied genetics section on, and so I'll look a little bit more at that. But that whole process of regulation, of going from the genome to RNA to protein, a lot of it, and what we know now is really kind of, this is where a lot of the research is now sitting, it's right in this level. It's the way we package our DNA, the way and this is where all of that memory, that transgenerational memory that article is talking about, is this modification of the way we package our DNA. Because it's a semi-permanent change. If we change the way we package our DNA, that's going to change what's accessible, what's able to be turned on and turned off. So it doesn't even matter if your gene is mutated or not. If you're not transcribing it properly because you have it packaged wrong, well, that's just as bad as if the gene isn't is mutated and not working as well. And so this idea of packaging and the way we can modify our histones and change the way the DNA interacts with the chromatin is a really important concept, and ultimately that's what like this transgenerational effects is all going to be housed right in there. So when we're looking at stuff at the chromosomal level, you got to remember that we're talking about eukaryotic genetics in this course, and so most of what we're talking about are diploid organisms. And so when we're talking diploid, remember we got 2N, so N is the number of copies of chromosomes you have. So you start with a sperm, you start with an egg, each are haploid, 23 chromosomes. They come together and make your zygote, and it becomes 46 chromosomes. That zygote goes through mitosis, 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 mitosis for its entire life. Mitosis, mitosis, mitosis in every cell type except for germline cells. So in every cell type, this is what we look at. For humans anyways, we have a diploid of 2N equals 46. It means that we have 23 chromosomes that come in pairs. These are our homologous pairs. And so remember, we have 22 autosomes and then two different sex chromosomes. And so we talked about different ways that, again, how all these can be rearranged. We talked about how we can use, this is skyfish, but how we can use fish to be able to probe regions on this, and I'll talk more about fish in just a second. So, this slide is so key to everything we did for the last 2/3 of the semester, okay? Because this is really what, at the heart of DNA replication, it's at the heart of RNA transcription, it's at the heart of most of the techniques that we've talked about, okay, in two ways. One, we have anti-parallel and we have complementary. Okay. So remember anti-parallel, we have our basic structure, our ribose down here, or a deoxyribose, so we're missing the oxygen. Remember how we number our carbons, carbon 1, carbon 2, carbon 3, carbon 4, carbon 5. On carbon 1, that's where we put our base, carbon 3, that's where we have that hydroxyl, and then carbon 5, that's where we put our phosphates. Now, it's critically important that we always have this 3' hydroxyl, okay, that's absolutely important for the formation of a new peptide bond between a new nucleotide. Now, this is what we're referring to through directionality. I know this is all review, but so we're going 5' to 3', the other side's going 5' to 3'. what's important is that this interaction of anti-parallel is going to hold true